In a previous lesson, we learned about the existence of orientation cells, or edge-detecting cells, in the visual cortex. We think they exist because of the nature of the wiring between the retina and the thalamus and the visual cortex, and they seem to be a first stage in visual processing. It's as if the brain is analyzing the visual world in terms of edges of different orientations. Of course, edges can be the boundaries of objects and their shadows. We also went on to try to conceptualize how neurons might come to represent more complicated objects. And we learned about a simple face cell, for example, might be getting inputs from different kinds of orientation cells at certain locations relative to each other. And the coincident activity of inputs would make this target cell respond maximally to a face. So we would call this a face cell. So we've got edge detecting cells in early visual cortex called area V1. And then farther along visual processing, we have cells that represent more complicated objects like faces and houses and things. In this lesson, we'll extend the story of the visual pathway as we travel from the visual cortex back here, area V1, down through the ventral part of the temporal lobe, so the inferior temporal cortex. So this is the major visual pathway dedicated to determining what kinds of objects are out there in the world. There is another major visual pathway that runs up into the parietal cortex, and that pathway seems to be processing where an object is in relation to the body. And so this area can compute how the body would have to move so as to interact with that object. But we're going to focus on this ventral pathway, what sometimes is called the what pathway, and the dorsal pathway is the where pathway. Now in this part of the diagram here, we're going to see the visual cortex back here, and these little marks here would be the orientation cells. And we'll see that here we have featured a target cell that seems to maximally respond to a triangle. So its preferred stimulus would be a triangle. And so just like the face cells that we saw earlier, we can imagine that this target cell is receiving inputs from a horizontal cell and maybe a diagonal like that and a diagonal like that in such a way that the cell responds maximally when all of its inputs are active at the same time, and that would happen when there is a triangle on the retina at that location, right? So this cell becomes sort of a triangle detector. Its activity would represent the presence of a triangle. Of course, we've seen that there are face cells, and the idea is there are cat cells and dog cells and cells that come to represent complicated objects in the world. So we start in visual processing with sort of edge detectors, orientation cells, and as the wiring proceeds down into the temporal lobe, we get cells with more and more complex preferred stimuli. Angles and object shapes and faces and animals, etc. Now one thing we'll notice as we think about what's happening to the flow of information is that we're going to see that what's happening is that there's a loss of some information to represent another quality, in this case shape. So as this ventral pathway is dedicated to representing the shapes of complex objects, we'll see that they lose the capacity to represent other features of the visual stimulus. So in this example, let's focus on a cell that maybe responds selectively to cats. Okay, so over here we're going to see what that kind of cell might respond to and what it won't respond to. So we're going to say that this cell is a cat detector, so it's not going to respond to giraffes or rhinos on the retina, but to a cat. So in this case, a lion. Let's say that's a lion there. But notice now in the second part here, the activity of that cell is invariant of location. So in other words, no matter where the cat is on the retina, if you're focusing here, if the cat were to the left of your fixation point, or if the cat were to the right of your fixation point or above the fixation point, that cell down in the temporal lobe here is still going to respond. So it doesn't care about the location of the cat on the retina. Neither does it care about the size of the cat. So it will respond if it's a small cat, a big cat, and a huge cat. Neither does it seem sensitive to the color or the motion or the texture of the cat. So in other words, as we move along this ventral pathway, cells become selective to a certain shape, but they don't care anymore about the color, the texture, the size, or the location of the object, just its shape. That's what we mean by loss of some information. The cells over here are not concerned with size, location, color, or motion. They just care about shape. Now that's in contrast to cells in V1, in the early stages of visual processing. Here the cells are sensitive to the size of the object or its location on the retina, because if the location on the retina changes, then a different population of cells will respond to that. And in addition, they respond differently to color and motion, etc. So what we see here is that these early cells are representing these qualities, but as we move down through temporal lobe, those qualities are lost so as to represent shape. So we can summarize this concept with these two diagrams here. For this first one here, notice we have the cat and the, whatever that is, some kind of bull or something. So some retinal images look somewhat similar, but they represent different animals, so they're different animals. Yet they fire many of the same cells in V1, so lots of the same cells will fire in V1 because they have similar shapes, but different cells in the inferior temporal cortex, right? So lots of cells in V1 will be responding to both of these. There's lots of overlap in the general shape here. But inferior temporal cortex, there will be some cells that selectively respond to the lions and some to this thing. Now over here, take a look at this one over here. We have, the big colorless lion and a small one that's at a different location and it's colored. So other retinal images are very different. But they represent the same animal. So it's a lion, but the images are different. So because the size is different, the location, the color is different, different populations of those V1 cells will be activated by these two images, right? So they fire different cells in V1, but the same cells in inferior temporal cortex. So as we move from V1 down into inferior temporal cortex, the cells become responsive to the object. What is the object, right? In a sense, the shape as a defining feature of the object, and not sensitive to the color, the size, the location, etc.